Hello, everyone. I'm Sanjoy Dutta, Chief Scientific Officer at JDRF. I look forward to this time of the year when all the hard work that all my colleagues at JDRF have been doing, along with multiple researchers and institutions around the world, supported essentially by all of you, to present these. So we've had another extremely exciting year. I was here a year ago presenting top advances in our fiscal year 23. Now I'm going to show you what is driving the research efforts and the development of therapies in this past fiscal year, FY24 here. So let's get going on this one here. I can move the slides. As you are familiar with, we work the pipeline. The pipeline is essentially a roadmap of going from very early stage discovery research all the way into therapies being in the hands of people with diabetes and them doing better. And we are proud to say that JDRF is the only organization that has the internal bench strength to support the pipeline, whether it's funding early research at the foundation or the T1D fund to translate these findings from benchtop and animal models into human clinical trials, proving that the drugs are safe, proving that the therapies are effective, then getting essential approval from the regulatory agencies in the United States and around the world. It's an important process, important step along the process, and then working with the payers, both private and public and federal payers, to ensure that the therapies are paid for. And then ultimately, the battle is not won there. You have to ensure that the healthcare professionals understand the value of the therapy. They discuss that with the people with diabetes, and they actually encourage and prescribe the use of these therapies. Only then will we achieve better outcomes. Another important point that we have all mentioned before, but I think it's worth repeating, is that this process, although shown linearly, is almost never a linear process. At each step of the way, along these chevrons, along these inflection points, you learn, and then you deploy that learning in a reverse engineering process to improve and enhance the previous step here. So it's more of a cyclical and iterative process, but for simplicity, it's shown here as a linear process going from left to right. As you are all very familiar with our JDRF research priorities, two main areas is our laser focus on curing type 1 diabetes, the mission for what we were founded. But we do know curing this complex disease will take time, and there will be multiple cures for the different people with type 1 diabetes. And in the meantime, we have a very robust effort in improving the lives, both the clinical outcomes as well as the quality of life of individuals living with this pretty debilitating disease here. In cures, we focus, of course, on how can we modify the course of the disease, stop it in its tracks to slow it down, halt it, or even reverse it, ideally, if possible. And there, in disease-modifying therapies, we have immunotherapeutic approaches for the autoimmune part of it and regenerative approaches to replenish the lost endogenous or internal beta cells, insulin-producing cells. To identify these individuals that will be eligible for the disease-modifying therapies, we will have to be able to screen or early detect these individuals. So there is a program here under disease-modifying therapies about screening the population. And I'll touch upon all of these with some examples in top advances. And then there's also another way, a very promising way of obtaining cures to provide insulin independence by using stem cell-based approaches to replace the lost beta cells within the body here. And in improving lives, we leave no stone unturned, whether they are devices that all of you are now experiencing and benefiting from. How can we have better devices, less intrusive, more automated, better outcomes, both clinical outcomes like glucose control, as well as less burden, disease burden, decision-making burden. And along with that, we know that there's significant improvements that can be made on the existing insulins. And I'll talk a little bit about this and other therapies that can be combined with insulin. One thing to note is almost all the non-communicable chronic diseases, type 2 diabetes, orthopedic diseases, blood pressure, cholesterol, and other lipid diseases, they all require what we call polypharmacology. There's no reason to assume that type 1 diabetes treatment should be only relying on insulin. And now we have a body of evidence suggesting additional therapies can add to the benefit that insulin alone can provide. And then our goal is to, of course, avoid long-term complications. If we can prevent it altogether, that's ideal. If not, at least we can slow the progression to diabetic eye disease, kidney disease, heart disease, so on and so forth. And then, needless to mention, this kind of a chronic disease requires 24/7, 365 management, and understandably brings with it significant mental health burden and psychosocial challenges. And that is as important as the clinical management of the disease. And how can we provide these supportive mechanisms and therapies that can address the psychosocial burden of living with the disease? There's significant progress in each of the areas, but I will have time to give one or maybe two examples in some of these areas. But most of them have been discussed by my colleagues earlier on in prior RIV calls, and I encourage you to go back and listen to them. A little bit of a 101 on biology here, just to level set everyone here. Disease-modifying therapies is exactly what the words mean, i.e. modify the course of the disease. And for this, we have to do at least two different things. It is an autoimmune disease, so you got to reset the immune process here to a pre-diabetes-like status. So one key component of a disease-modifying therapy is rebalancing or resetting the lost immune balance, where your immune system is now hyperactive, destroying your insulin-producing cells. The second complementary component of it is regenerate or regrow or replenish the lost beta cells within the body here. There are ways to regenerate the beta cells in the pancreas here. This has been a very challenging area of research, but nonetheless, we are pursuing this combinatorial approach of addressing both the immune system and the beta cell, and that's what defines disease-modifying therapies. I'm really pleased to report that in prior years and in FY24, we have significant progress in the disease-modifying therapy space here, and I'll give you two examples in the next slide here. So, as I mentioned, multiple therapies, and most of these have been tested in what we call stage 3 type 1 diabetes. Stage 3 is when people have been put on insulin treatment. They still have some remaining insulin production as measured by a surrogate marker called C-peptide. C-peptide is essentially a part of insulin that gets clipped during its production within the body here. So measuring C-peptide provides a very steady measure of steady surrogate or proxy for what the insulin production internally is. And this is kind of the marker that we are pursuing here. So in two different trials, phase 3 trials done this particular year, one is teplizumab that you're all very familiar with. Now, just a reminder, teplizumab or Tzield was approved by the FDA in 2022, November 2022, for stage 2 type 1 diabetes. These are people that have islet autoantibodies, so they have an active immune system, they have glucose intolerance. In other words, they're not able to manage the glucose rise after a meal. And so they are progressing towards a state of insulin requiring diabetes or stage 3. So Tzield is now approved for stage 2. This particular trial, sponsored by Sanofi, was conducted in over 300 individuals, children with stage 3 type 1 diabetes, using teplizumab. And what's shown on the top right panel is the people that got the drug, i.e. in the solid blue line, versus the people that got the mock or the placebo in dotted blue line. You see a differential between the levels of C-peptide. Now, a reminder, as I just said, C-peptide is a proxy for amount of insulin your internal cells are producing. So the higher you can maintain C-peptide, it means the healthier your beta cell population is. And it's quite clearly demarcated here. The people that received Tzield were able to, at the end of the 78-week period, which is about a year and a half or so, 78-week period, were able to retain more beta cell production, insulin production from beta cells compared to the placebo group. Very similarly, in the bottom right panel is another clinical trial that we supported, JDRF funded, both JDRF-I and our affiliate JDRF Australia funded, with a therapy called baricitinib, which is an inhibitor of another protein called JAK. And then this particular therapy is actually approved for other indications, but not for type 1 diabetes. A lot of prior work indicated it could work in people with type 1 diabetes. So we had the conviction that this is something that we need to test here. And this was also done in stage 3 type 1 diabetes. i.e. people newly diagnosed with type 1 diabetes. And this was done both in adult population as well as in children or below 18 years of age. And again, on the bottom right panel, the solid blue line are individuals treated with this therapy called the JAK inhibitor or baricitinib. The title's acronym is BANDIT. And in dotted blue line are people that received the placebo. And you see a very clear demarcation of retention of insulin production in the body with baricitinib versus without. These are very promising. As I said, I'm presenting results from two studies or two different therapies in FY24. We have had one more therapy that reported out and many more that are ongoing and hopefully will report back in the coming months or over the next year or so. Now, if stage 3 type 1 diabetes, these are the therapies being pursued here, why are we not seeing more of these? Why are we not seeing them quicker? Why is there still not as much therapy development as we would like to? And we think one of the major barriers is the current regulatory status for these clinical trial endpoints has become a bit challenging. Just to prove, this is a summary slide of a lot of work done by JDRF staff, particularly my colleagues Esther Latres and Mariana Marinak, but also many, many other staff members, as well as multiple investigators outside of JDRF. And this is data for over 20 years that have been summarized or evaluated here. On the top, you see, top left, you see where do the regulators stand right now. And there is some differential between US regulators and European regulators, but the bottom line is that C-peptide, which we believe is a bona fide marker of beta cell health, has been accepted by different degrees by different regulators, but still at the end of the day, they're not validated as the surrogate endpoint to use for drug development. Just like we have hemoglobin A1C, it's a surrogate for long-term complications, and you can get diabetes therapies approved using hemoglobin A1C as a marker, a surrogate marker, a proxy. Here, we think C-peptide should be used here, but this is where the FDA and the EMA stand. And so what we did is we looked retrospectively at evidence for this C-peptide as a validated surrogate marker that can predict a therapy outcome and its potential long-term clinical benefits down the road, which is impossible to show in a 1-year trial or in a 2-year trial, especially in a very aggressive disease here. And so this is our next goal, is to bring this to the regulators to show them that this is the evidentiary basis to validate or approve C-peptide as a validated surrogate endpoint. And then in parallel, and kind of related to it, is because of a lack of this guidance around this pathway here, the perception and sometimes the reality is you needed to do very large trials for a very long duration of time and with a lot of people in the trial. All of these are prohibitive. You don't have that many people. The disease is fast progressing. You cannot show a change in hemoglobin A1C in such a fast progressing disease. And it's very difficult. So one other effort that the JDRF team pursued here is, can we use modern technologies such as modeling and simulation tools to learn from prior trials? So about 21 clinical trials in stage 3 type 1 diabetes were analyzed here to now show not only what is shown in the above panel, C-peptide as a validated endpoint, but also develop a model or multiple models for smart clinical trial design. By smart, we mean you don't need 500 people per arm, placebo and drug. You don't need 5 years to do the trial. You don't need hundreds of millions of dollars to do the trial. You can do the trial both in pediatric and adult population with a reasonable number of people, like 60, 100, 150. You can do it in 6 months' time. You don't have to go for 5 years. And hopefully, in the months to come, C-peptide could be the surrogate endpoint approved by regulators to use in the trial to show the potential for long-term benefit. We believe that these two are seminal work that will change the field and will incentivize further investment by multiple pharmaceutical companies in this space here, and we will have definitely moved the needle there. So I intentionally spent more time on this slide because it it is kind of it's kind of a landmark approach right now that will really move the field forward. A couple more examples. I spoke to you about clinical trials, right? And we know very well that clinical trials are happening today because there was promising early stage discovery work done 5 years, 10 years, in some cases 15 years ago. So discovery research is critical for establishing the framework and the groundwork for tomorrow's discovery. So what's shown here are two very basic biology concepts that we have funded over the last couple years or so, that look quite promising and definitely interesting. To the left panel is an investigator in Australia that had an innovative grant just to prove a hypothesis using therapies that are now used in cancer biology, inhibiting a particular enzyme called EZH2. I won't get into the details, but this particular enzyme allows the modification of DNA. And what happens by that is cancer cells can no longer multiply and propagate, so it stops it in its tracks. The same concept can be used to our benefit, actually, in type 1 diabetes, where inhibiting this particular enzyme can allow the cells, the stem cells within the body, instead of promoting their proliferation, can convert them into insulin-producing beta cells. So this was actually a kind of a shotgun approach, and it worked. And in its preliminary evidence, mind you, this is early preclinical. However, the results are interesting enough that we are now funding further expansion of the study and validation of the concept that this particular inhibition of this particular enzyme can have legs in future development of a therapy for internal regenerative medicine for beta cell insulin production. On the right panel is actually the investigator that led the baricitinib study that I just spoke to you about, Professor Thomas Kay, again in Australia here. A big question in biology here is when you have an infection, when you have cancer, you have active T cell components, just like you do in an autoimmune disease like type 1 diabetes. But over a period of time, repeated infection or other cancer, these T cells demonstrate what we call exhaustion phenotype. They get tired, they give up. And that's when you get remission from the disease. Of course, there are other complexities and other treatments I won't get into now. However, in autoimmune diseases, definitely in type 1 diabetes, in our disease here, there does not seem to be any exhaustion of T cells. So they relentlessly keep destroying the beta cells. So even if there is any internal production of beta cell or beta cells injected from outside, such as in transplant medicine, these T cells will go and destroy the beta cells again. So what Professor Kay did is he amassed a team of multiple experts with a lot of diversity in their knowledge and experience, such as cell biologists, geneticists, and many other expertise, and combined them to sit in a room and think about understanding what leads to this inexhaustive phenotype of T cells in type 1 diabetes, and how can we devise therapeutic approaches to induce T cell exhaustion and thus provide a stop to the disease, or in some cases, ideally, to even reverse the disease, but that might be a little bit further down the road. So these are exciting projects. We don't have data today, but in the coming years, we will definitely know if these pathways work. And if there is promise, then we will definitely take it to its next inflection point, which I showed you in the pipeline at the beginning of the talk. Moving on to, and continuing with the cures theme, but moving on to a very promising area that you all have heard a lot about is cell replacement therapy. So again, a very quick biology one-on-one. I was so far talking about creating or regrowing or replenishing your own beta cells inside your own body, or regeneration. Here, I'm talking about replacement. Here you take a source of cells from another human being and you multiply them and convert them into insulin-producing beta cells outside the body, in a petri dish, flask, however you do it. And then you put them back in the body. That's the concept of cell replacement therapy, often known as transplant medicine. However, we know, because the body's active immune system, our immune system, as well as the fact that you're getting a foreign supply of cells, both your own immunity, autoimmunity, as well as your immunity to a foreign agent, these cells will destroy the cells. So the second part of this whole portfolio, and an important part of it, is to find protection strategies. So make the cells and then protect the cells when you put them back in the body. We've had a leadership position, and probably we are the largest funder, not-for-profit funding organization here supporting cell replacement therapy. And we are very pleased that multiple commercial investment opportunities have sprung up over the last 5 or 6 years or so, and they are playing in our disease space. And I'll give you 3 or 4 examples over the next slides. Now, if I was in person, if I could see you all, I would take a show of hands of who hasn't heard about Vertex, and I bet that there would not be a single hand up here. So I'm not going to introduce Vertex here, but provide you an update. They are pursuing 2 clinical trials with the same source of stem cells that Professor Doug Belton had developed that we funded, and then the T1D fund invested in the company SEMA, and now Vertex is pursuing a cell replacement therapy using VX-880, where there is full immunosuppression, just like you would do in a pancreatic islet transplantation from deceased donor islets. And then the second trial, they're using the same cells, but now in a protective encapsulation strategy, and that trial is also ongoing. We don't have an update on the second trial, the VX-264. I'm sure we will have an update very soon. But the VX-880 trial that showed very promising results in many of the people in the trial showing insulin independence with just 1 dose of cells. And they, however, experienced, unfortunately, deaths of 2 of the patients due to unrelated causes, and it was put on a regulatory, what we call a clinical pause or a clinical hold. We are very pleased to know that after a thorough review by independent monitoring committees, that the hold has now been lifted and the trial is ongoing. So that bodes well for all of us here. So more to come in the coming months. And this is a very promising area, certainly spearheaded by Vertex now, but there are many other companies and academic institutions pursuing it. To the right of the slide is what was actually approved at the end of FY23, but we didn't get to report back to this particular group of our volunteers here. The company's name is CellTrans, and what the product's name is actually quite a mouthful. It's donislecel, like teplizumab, it's donislecel, and the trademark name is Lantidra. So what they're doing here is they are using cadaveric donor islets or deceased donor islets, and they have obtained regulatory approval, what we often technically call a BLA or a biologics license application from the FDA to use this as a therapy in people with type 1 diabetes and in a subset of the people with type 1 diabetes. This is actually, although we know that islet transplantation has been ongoing for a number of years and is also ongoing in different parts of the world, we did not have a regulatorily approved product in the United States. So this is important just at the level of a breakthrough product that has made it to the market. But more importantly, what I and my colleagues see is this also sets the stage, because this first step of breaking ice is very important to understand what is required to get a cell replacement product approved by the regulators. We're hoping that many of the lessons learned in this will apply to Vertex and many other companies trying to pursue cell replacement therapy. We understand the products are very different and will have different requirements, but at least it sets the stage to something I'll discuss in the last slide about clinical trial guidance. Moving on to 2 more exciting examples. As you all know, we are now doing cell replacement therapy either with full immunosuppression, just like islet transplantation, or using encapsulated strategies, as I just showed you from Vertex, and in the past from ViaCyte. But our actual, to realize and fulfill our dream of a cell replacement therapy is actually a cell replacement therapy product that requires neither any encapsulation nor immunosuppression, or in other words, you have now smart cells going into the body, smart beta cells going into the body. And one way to approach it is to modify the genes that confer it this immunity against a body's immune system. In other words, they are cloaked, they're hidden from the body's immune system, or they're immune evasive or hypoimmunogenic cells. You'll hear a lot of technical terminologies, they all mean the same. We have a very, very robust effort in approaching gene editing methodology, in utilizing gene editing methodologies to generate beta cells that are invisible to the body's immune system. We have these centers of excellence and plus other efforts outside the center of excellence. We were very pleased to see most recently a mid-sized biotech company called Sana Biotechnology invested significantly in our disease, and they have actually implanted genetically modified cells, islet cells, into a large animal model, what we call non-human primates or monkey models, and they have achieved insulin independence without the need for any immunosuppression, any encapsulation for, I think, 6 months or 8 months now. That is very promising, and they are pursuing a clinical, they are considering pursuing a clinical trial in type 1 diabetes. So it's actually super exciting to hear, different types of approaches going on, and all of these are grounded in our cell replacement strategy, and we are independently funding a lot of these efforts that are in turn helping the companies move forward here. And I can go on, but this will be the last example in the cell replacement therapy area. Very recently, as recent as I think yesterday, it was announced by this small biotech company called Eledon Pharmaceuticals that our T1D fund is invested in providing alternative approaches to immunosuppression. So right now in transplant setting, whether you do deceased donor islet transplants or you use VX-880 like stem cell-based islet transplantation, what you do is you do a whole body flushing with immune suppressants. And that's the only way to protect these cells and ensure their durability in response. But whole body immunosuppression is A, unnecessary, and B, not really very safe. And in the long term, people do end up having side effect profiles. In some cases, it's mild. In some cases, it's very severe. And so that has been a deterrent in the process of expanding deceased donor islet-based transplantation procedures. What we are experimenting now, and JDRF, our research team is now funding a clinical trial testing the same agent, which is anti-CD40 ligand. It's an immunotherapy, not an immunosuppression agent. It's a specific immunotherapy agent. It's an antibody, and trying to see if this antibody is able to elicit the required immune protection to the cells rather than bombarding the whole body with immunosuppressants. Interestingly, the same compound was donated by the company Eledon Pharmaceuticals to the academic trial that we are funding. And in parallel, now the T1D fund has invested in the company. So more to come in the coming months, but this is super exciting. The technical name of this anti-CD40 ligand is on the slide. Again, it's a mouthful. It's called tegoprubart. But more importantly, it's an immunotherapy approach, not an immunosuppression approach. Now, we know that a lot of people are being affected by type 1 diabetes. Unfortunately, the incidence and the prevalence of this disease is on the increase. And we know that different people in different ages are getting it, different ethnicities are getting it all over the world. And as we identify these people, we're trying to prevent DKA in them. We're trying to ensure they have access to clinical trials if they're willing and able to participate. So we move our disease-modifying therapies forward faster. However, one thing we often overlook as a community, and we haven't done justice here, is that we talk about screening, we talk about risk detection, we talk about early detection, we talk about antibodies, but that is not enough. That is a tool, that is a marker in the blood. But actually what prevents DKA, diabetic ketoacidosis at diagnosis, what actually makes a family ready for the ultimate diagnosis of T1D, what actually allows for preventive therapies to be tested in these people is monitoring. Currently, we don't have any monitoring guidance. Different parts of the world, academic institutions have their own protocols for monitoring. Now, the research institutions that are well known are able to do this. It's a very complex process. What GDF wanted to do is we wanted to lead the field forward and address this problem in a simple, consistent manner. So there is a guidance that is accepted by the diabetes societies and that provides kind of a roadmap or GPS to clinicians to be able to use this. So we developed this guidance document. In the next slide, I'll show you the number of people it was required to corral the entire process. And I want to give credit to my colleague, Dr. Anastasia Albanese O'Neill, who led this process from the JDRF side here. I was there along with her throughout the process, but she really did a marvelous job here. And this guidance document has all populations, pediatrics and adults, provides education for both the healthcare provider and the families affected, and important psychosocial considerations, because getting this information about a diagnosis of autoantibody positivity and knowing the sequelae of what is imminent down the road is obviously extremely mentally burdensome and sometimes pretty helpless, both to the families and the healthcare providers. So all these recommendations were reviewed thoroughly by clinical committees, and they were based on the level of evidence available. And I'm really pleased to say the punchline is in the bottom right, is that all organizations as of 2.5 weeks ago have endorsed the guidance document in a record time of 1 year. I don't think there's precedence for this, at least in my career. And there will be a scientific session at the ADA in Orlando next month. In case you're planning to attend ADA, this is a plug for you to go to the JDRF session on Monday morning. I forgot the date. And this is just some of the behind the curtains work about 73 international experts in what I showed you on the previous slides: pediatricians, pediatric endocrinologists, adult clinicians, adult endocrinologists, psychologists, so on and so forth, you name it. In 4 continents, 8 partner organizations listed at the bottom of the slide, led by JDRF, we were able to get this guidance document prepared, and now it's been accepted by the major diabetes organizations and will be published in the near term, in about 6 to 8 weeks' time. So we will communicate around that when the time comes. But in the meantime, this has been a huge achievement in FY24 that we are all very proud of. To finish up the rest of the talk, let's go into an important area that we have been equally passionate and successful in improving lives. In improving lives, again, just like disease-modifying therapies, it is what it means. It improves the lives by providing good outcomes, clinical outcomes, and also good mental health outcomes. And they could be achieved by different therapeutic means. It could be a psychological intervention, like a behavioral therapy. It could be a drug. It could be advanced insulins. It could be device-based approaches. It could be a combination of the above. Just 3 slides on some examples here. You're probably most familiar with this slide here about the growing, thriving ecosystem of advanced, more advanced, even better devices, smaller, easier to use, longer lasting. I won't read out each of these, but in FY24, at least a couple of these were approved, and we were very excited about it. What's more important to know is that this is all the dividend that we are getting from the investment we had made, you had helped make, and you probably contributed significantly to this 20 years ago in 2005 when the JDRF Artificial Pancreas Program was launched. Funding of research, regulatory pathway, payer pathway, education of the community, both healthcare provider and patients, the benefits. And now kids as young as 2 years of age and sometimes even younger can avail these therapies all the way into a geriatric population. And so I don't need to expand further on this. We do have other mechanisms within our research portfolio to even further advance this, but we're really happy to say that this is an ecosystem that's thriving a lot due to our leadership and past funding. There are niche things that we can do, and our team will present them to you in the next fiscal year over an update on what we are doing in the device space now. But our role is very focused into some very niche products that we can help move forward in an accelerated manner here. We have been not only extremely focused on what we call adjunctive therapies, or therapies that can be accompanied along with insulin to provide added benefits, very critical benefits. I'm pleased to say we're not only have been leading this field, we sowed the field. I believe we are probably the only ones supporting this field in a major way right now. Whether it's research funding, whether it's partnering with stakeholders, whether it's getting a regulatory pathway there, and then eventually, when approved, hopefully discussions with payers, should there be a need or any roadblock there. There are just 2 examples shown here, and I alluded to this earlier in my presentation about polypharmacology, about treating type 1 diabetes as almost any other chronic disease that you require multiple therapies to be able to provide the best benefit possible. The benefits could include better glucose control, metabolic control beyond glucose numbers, such as lipid, body weight, so on and so forth. And then eventually, as we are seeing now, a growing body of evidence and approval in a non-type 1 population for kidney and heart disease. To the left is shown a once-weekly injectable class of drugs that is becoming a miracle drug now. You all have heard about this, whether it's Ozempic, Mounjaro, Zepbound. They are different drugs belonging to the same or similar class of what we call GLP-1 agonists. And now there are dual and triple agonists coming out or already out. And on the right is the oral tablet, inhibiting a particular class of transporter proteins that helps eliminate extra glucose from the blood through the urine here. And the example given here is made by the company Lexicon. It's sotagliflozin or Inpefa here. What are we doing here? A lot of things here. The main role we are trying to do here is what we are very good at. Try to get the body of evidence that these therapies also work in people with type 1 diabetes. There's no reason to believe it won't, but we need the data to prove it, because it works in people with type 2 diabetes. It works in people without diabetes, but kidney disease or heart disease. And there's no reason why it shouldn't be working in type 1 diabetes, but nobody else is pursuing it in type 1 diabetes. Most of the commercial investors, excepting probably one, are not investing in type 1 diabetes at the present time. And so we need to fill both the funding gap and the data gap and play a leadership role, just like we have done in some other areas that I presented earlier in this presentation here. So we're pursuing some of the usual suspects you've heard about a lot, GLP-1 class, SGLT class, as well as other classes called a mineralocorticoid receptor antagonist, which is not shown on the slide here, for kidney disease, for heart disease, and certainly for improved glucometabolic control, glucose control and metabolic control. In the middle panel, the devices continue to work miracles. We have shown that these devices work in different populations, different age groups. We have shown the benefits. We have shown in the past it works in pregnancy. It has changed standards of care in the United States and around the world. This is another study showing that this approach, how specifically it helps the women that were pregnant and experienced poor outcomes, but when put on these automated insulin delivery systems, formerly known as artificial pancreas systems, how they're getting better glucose time in range, how they're getting fewer hypoglycemias. They're not gaining as much weight because of the tight control here. And the outcomes were also translatable to the newborn babies here. So it's an extension, but important findings with the advanced systems here. And to the right is actually a very cool study. It's a very cool study. You know how we know that glucose control is critical to avoid complications. We've used hemoglobin A1C as a marker of that, and all of that has been the gold standard. We believe that time in range using CGM measurements can also be a very good marker for predicting long-term complications. And this is a study in which one of our investigators did a retrospective analysis of data sets and showed that for every 5% decrease in time in range, so poor glucose control, you actually saw a 16% increase in diabetic eye disease. While the data is not shown here, I bet the same is true in kidney disease as well. In other words, our constant, fight in the field to get the data, to impress the regulators and convince them that time in range is a good marker of near-term but also long-term benefits in people. This is actually a good evidence to show that increase in time in range will decrease complications, at least of the eye, or the reverse here. So this was a very cool study done in a very short time frame and in a very small budget here. Now, just to bring us home here, all of these can be possible with a lot of research funding, a lot of advocacy efforts, a lot of volunteer leadership, certainly the staff leading the way with the strategy, other stakeholders. But at the end of the day, we can only do so much. These clinical trials still today are difficult to recruit. They're prohibitively long. Many people withdraw after signing up or halfway through the trial. So we need to ensure that the clinical trials are progressing fast so we can get results fast. we can get to therapies faster, or we fail fast, and then we don't throw good money after bad. So clinical trial participation, and I would posit, can be your biggest volunteer activity. Certainly, we need support, funding support. We need other kind of policy support and everything else from you all. But clinical trial participation is probably, you're helping yourself, you're helping the community, you're helping our mission to move forward here. And these otherwise will become even more prohibitively expensive and lengthy, and these results will actually deter further investment by key stakeholders in the field, such as the pharma and biotech sector. We have tools, and we mentioned this in almost all of our presentations because it is so important here. We've had a clinical trial matching tool now for 7 or 8 years now, and now we have it in English and Spanish. This is a matchmaking tool. Use it. It's quick, it's easy, it's under 60 seconds, and it does exactly what it touts to do, matches. And after that, you can go on a date with the clinical trial site and determine if they're the right, if that is really your cup of tea or not. And then to the right, we constantly provide these updates in the pipeline or the research newsletter, letter that is led by my colleague Brian Herrick, who also leads the RIV program here. This is a very rich source of, in a nutshell, advances each month here, and includes at least one, sometimes two, clinical trials that we have either just funded or require some information or whatever it is. I encourage you to read this if it comes into your inbox. And if it doesn't, please ask us. We'll make sure that you are getting it, because I find this to be a really quick cheat sheet and a one-stop shop of what happened over the last 30 days' time here. Bring you back to the start, back to the future here. I started with the pipeline. I started with our bench strength. I started how we help move the therapies along, research along, these critical chevrons and inflection points between the chevrons here. Each step is important. It's an iterative process. It's more often circular than linear. But at the end of the day, this is also the metric by which we measure our success. Are people with type 1 diabetes doing better or not? So hopefully you were as excited as I have been over FY24. Now, what to look forward to. This is my last slide, and it's just a subset of what we are all looking forward to bring to you. I talked a lot about clinical trials. I talked a lot about progress there. I talked a lot about our barriers there. So one of the things the teams are doing here, research, advocacy, the T1D Fund, is putting our heads together to create clinical trial guidelines for cell replacement therapy, for disease-modifying therapy. I showed to you earlier a monitoring guideline that is now going to become the gold standard once because it's been endorsed by the diabetes societies. We're also working on a screening guideline, more to come for that in the future. But what we've already started working is on clinical guidelines that can accelerate cell replacement therapy and disease-modifying therapies. We desperately need that. There's another important area in cell replacement therapies. We have been supporting, in a small way, a beta cell factory. So there are more cells available for people to do research, to people to develop protection strategies. We're going to significantly ramp that up because that seems to be a barrier or a roadblock or the slower part in the wheel here. And then just like we showed you on the slide earlier, if you want to go back and look at it, the data on the C-peptide and the modeling of the clinical trial, we're trying to do the same thing for stage 1 and 2. That was for stage 3, newly diagnosed type 1 diabetes. We also need to do for stage 1 and 2, because otherwise, again, the same thing. If you don't have an endpoint that is quick, that is accepted by the regulators, these trials are going to be prohibitively long, and it will become very difficult to prove whether a therapy is working to prevent or slow the disease or not. On the right, a lot of excitement. I spoke to you about the trials we're doing in glucometabolic and in complications. We're expecting at least a subset of them to read out in this coming year, and more to come on the device front, not shown on the slide here. And we have a very cool RFA that we are going to make a funding decision subject to governance approval next month at the end of the fiscal year on key, not only psychosocial interventions, but implementation of this. Because it's one thing when you take the horse to the pond, but it's another total different tactics to make the horse drink the water. And this RFA was directed towards that implementation science of implementing an intervention, such as a cognitive behavioral therapy or mindfulness therapy or something else. How do we intervene it in our healthcare system to ensure people are actually getting the benefit from it in a sustained and consistent way? So I look forward to meeting you all again in future RIV calls. My colleagues will be presenting, but I am super excited with what has been achieved in FY24, but we have not cured the disease. So a lot more to do in FY25 and beyond. So stay tuned, and I will be happy to take questions when you have them either online or in future calls here. Thank you very much.